Hi everyone.
Hello.
How are you feeling, Clyde?
Um, I am not well. I
Are you
got
okay?
sick last week,
Oh no.
which is it's a wonderful time to be sick, but yeah, it's fine. I'm working through it. I have my tea here.
That's good.
Good morning, everybody.
Good
Morning.
morning. Wait, my camera still isn't working. What the heck?
So, so Clyde, I hear you're not feeling well.
No, I got
Sorry to hear.
some kind of bug or something.
Yeah,
Hold on, my camera is not working. Sorry. You can
yeah.
continue. Let me just fix this way.
And it looks like we have a special guest here, D Black. Uh,
That would be my mother.
that's your mom. Okay. Hi, Clyde's mom.
Hi, I just got my microphone to work just in time. Hello.
Good, good. And you're joining us from Edinburgh, I guess, right?
I'm in Edinburgh, yep. Ooh.
It's a great town. Love that town. Yeah. Good. Well, welcome. So we're a
Thank you.
small group and we've been enjoying working with Clyde. Um, and this is his defense of his honors thesis, right, Clyde?
That's correct.
Good.
It's gonna
Okay.
be half thesis and really just kind of half project update because this project is moving forward, um, and so there's a lot of future directions that I'm still planning.
Good. So, well, Clyde, I'll just turn it over to you then, right?
Yes, I would like to get my webcam working,
Okay.
but I just cannot for the life of me.
I guess the important thing, can we see your screen or
Oh yeah,
can you
yeah,
see
that
your,
that'll work fine.
uh-huh.
But it would be nice if I could this work. Alright, I don't think it's going to work. I
Okay.
tried reconnecting and then it asked me specifically to use the camera and I said yes and then it said it couldn't. So
Oh, oh well.
Okay, Clyde. Well, can you share your screen? Why
yeah,
don't you share
yeah,
your
yeah.
screen so you can see what you got there? Good.
Alrighty. So my presentation is titled Group Living and Stress Response in a Colonial Green Alga. And I think we will start with a quick refresher of evolution and the history of life. All life is descended from a single, simple common ancestor, and throughout evolution has acquired a great diversity of complexity that we see today. The tree of life is amazingly diverse, and all of this came from heritable variation in traits, which was promoted by natural selection based on fitness. Fitness acts upon individuals that have heritability and variability, and all that together is what we call evolution. The unit of selection that fitness acts upon is what we call an individual. But what an individual looks like has changed over the history of life. The earliest individuals were molecular replicators, and eventually they evolved to influence each other in a confined space. Some spaces of replicator networks did better than others, and eventually the networks became the individual, where fitness is not the same as the combined fitness of replicators inside of it. This process is called an evolutionary transition to individuality, and it represents a fundamental jump in the complexity of life. There have been many evolutionary transitions in the history of life, and there are many levels of complexity created by them. And greatest hits include the genesis of the cell, eukaryotic cells from prokaryotic cells, multicellular individuals from unicellular individuals, and eusocial colonies from individual creatures. Transitions to greater complexity like this are rare in the history of life, but are some of the most interesting and important events to ever occur. The evolution of multicellularity is a popular and important transition. Multicellularity has evolved about 25 times in eukaryotes, whereas, as far as we know, earlier transitions like the eukaryotic cell itself have only ever happened once. A multicellular organism has access to a lot of complexity, such as organs and tissue, the ability to shield reproductive lines from damage and stress, and access to a macroscopic body, which isn't entirely exclusive to multicellular organisms, but it's pretty rare otherwise. There's a lot to gain from being multicellular, although, granted, I am a bit biased because I am multicellular, and I think I get on pretty well thanks to all the complexity. In the evolution of multicellularity, there are traits that an individual or a group of individuals acquire to become a committed multicellular organism. This starts off with just the ability to be colonial and live in a group in the first place, and then this group gains things like evolution of cellular differentiation for division of labor, conflict mediation mechanisms, one of which is the division of the labor of reproduction, so reproductive specialization, and generally increased body size. As these traits are acquired, fitness moves from one level to another, although importantly, fitness still applies at all steps during the evolutionary transition, and in the intermediate phases, the collection of individuals and individual itself experiences fitness at both the cell and group level, and this is what we call multi-level selection. I am particularly interested in early multicellularity, that is, the first stages where groups are just forming, but they don't have complex conflict mediation mechanisms to lock in that style of life. Early group living tends not to persist. It tends to be a somewhat unstable state. Usually cells will either regress and go back to just a unicellular lifestyle or gain more traits and become more stable as a committed multicellular organism. There are some extant individuals, though, that live in this kind of transitional early group stage, and Gonium pectorale of the Volvocene algae is a good model organism that we can use to study this state. It is a kind of transitional genus in between Chlamydomonas, which is a unicellular algae, and Volvox, which is a committed and differentiated multicellular algae. And what we're interested in here is how Gonium modulates the costs and benefits of group living to maintain this early non-committed state. The study organism we're looking at, Gonium here, has condition-dependent expression of the group phenotype, and this is important. The excuse me. The group phenotype can be either expressed or not expressed depending on what is going on. So by default, it is expressed. Gonium is a multicellular organism, but under some conditions, known particularly density stress, the Gonium will dissociate, where cells break off and start living on their own. It's unknown if this dissociation is a cell-level adaptation, where the costs are too great in group living, and so this lifestyle breaks down in favor of a single-cell lifestyle, or if this is actually a group adaptation in and of itself to do something like propial dispersal. In particular, for this work, we are choosing to use the strain of Gonium pectorale K3F34, which is a minus mating type with an NIES code here. And this is because it has good expression of this kind of partial multicellularity under our conditions, and has also been sequenced previously and is kind of a standard strain used in this kind of research. The inspiration for this project is based on some work that Deborah Shelton did in the lab, which showed a dramatic change in Gonium group living as growing time and density increased. Moment. Take a lozenge. So here is this experiment summarized in a graph here. At 7 days, the culture is primarily multicellular, where by 14 days, it is gone primarily unicellular. We can see here on the microscope slides that where Gonium is living in groups under non-dense conditions, it is gone into primarily single cells under dense conditions, although some colonies do still persist. And this is interesting because it indicates that density stress induces a shift in a major life history trait, that is, being multicellular. So we wanted to tackle this project with a new tool set, in particular a machine called the FlowCam, which allows for automated capture of thousands and thousands and thousands of particles, whereas you could only fit so many onto a hemocytometer to count them traditionally. This makes it possible to image a significant portion of a population. We put in 0.5 milliliter of sample and can get almost 30% of all of the particles in that sample captured by the computer, which is a lot more than you could get with traditional microscopy. And having all of these images be digital and automatically captured makes it theoretically possible to do automatic categorization. None of the data in this presentation was categorized automatically, although it is an implementation that we're working on. So the big picture question that I want to answer here is, what does the organizational behavior of Gonium pectorale indicate about how group living is preserved during the evolution of multicellularity? And to get specific about what we're doing here, how does Gonium pectorale modulate group organization in response to environmental costs and benefits? The experimental approach that we're using to look at this is to describe when Gonium dissociates. So, as previously seen, it seems to dissociate at high population density, but we're going to get a little more specific about when it's actually happening. And to do that, we're testing the phenotype response, phenotype in this case being multicellularity, to density stress. We grew separate flasks to high density and tested 3 points in the life cycle. The rationale here is to look at the different points across the life cycle and see if we can see when Gonium is dissociating within that. And we're using the proportion of multicell phenotype in the population imaged as our statistic here. So we predict that as time and density stress increase, we're going to see a greater difference in the multicell proportion from when the Gonium reproduces and hatches into a colony to growth when we think it will dissociate. Here's the experimental figure for what we ended up doing. I won't go too much into this, but basically just to walk you through, we have a density-controlled inoculation where we are making several flasks to grow in parallel. These flasks were then grown in 2 incubators under the same conditions that were offset in time so that we could sample them closer together and still get that full range of time points. The life cycle here is shown on 2 24-hour clocks. And then when we do sample, we take 3 technical replicates and put them through the FlowCam with these settings and do phenotype categorization to find population statistics. There was also a molecular component here, which is not my project, so I won't get too much into that. On the bottom here, we have the same life cycle time points set up to show at which point in the Gonium life cycle they are occurring, and in real time, the generational difference of what's going on in the populations. And then we did this entire experiment 3 times to reduce stochasticity. The results for the density stress experiment are summarized in these 2 graphs. So we have the multicell proportion of the population at the 3 life cycle time points for each of the days that we measured, which is 2 to 14. And then we also have a graph of multicell proportion compared against the population density of the flask as calculated by the flow cam. There are some issues with using the particles in the flow cam, namely that, especially under high multicellularity, it's just counting the images that it sees and not the amount of cells in them. But it's pretty good for what we need it for right now, and we also see this decrease in multicell proportion as the density increases. So we see a notable decrease in multicell.
Clyde?
Yep, go for it.
Could you just a point of information? Could you just tell us a little more about the life cycle time points and
Yes,
what the thinking is there? Yeah.
I have a slide for that, which didn't really fit into my story, but we can look at it here. So here is the Gonium life cycle, and this is grown under a 2-to-1 ratio of light to dark. When a colony is born, that's around time point C, it starts to grow in the light, and it may dissociate, but it grows larger and larger. And then at the end of life phase, it will decide whether or not it's going to reproduce, whether or not it's grown enough to reproduce, and that is the time point A. If it has decided to reproduce, either as a single cell or as a group, it will then start to divide within itself, make divisions, and over the dark phase will form daughter colonies, which is around time point B. So one cell can form a daughter colony, or in a group of cells, all of the individual cells in the colony will form a daughter colony. So that's shown well here in B and C. Here is an 8-cell colony dividing as a group. So each of these used to be a cell and is going to become a daughter colony like this. All good?
Thank you.
All right. So we see a notable decrease in the multicell proportion with increased growing time and density, which tells us that group living is sensitive to the conditions present at high density, as shown by Deborah's experiment. Although we do note that colonies persist at low concentration even under conditions which they are sensitive. So this curve kind of levels off here at about 15% multicellularity, even under high density conditions. Another thing that this decrease in multicell proportion shows is that dissociation is a cost mitigation strategy. The stress and cost at high density is having some effect on Gonium, and so it is dissociating to deal with that cost. The life cycle point does not appear to have a big effect on the multicell proportion. So unlike we predicted, there's a pretty inconsistent relationship between the multicell proportion at different life cycle points. And so we cannot support that the reduction in group living is driven primarily by intra-life cycle dissociation. That is when a colony is born and then dissociates during the light phase. Instead, we think that the decrease in multicell proportion we're seeing is likely much to do with quiescence, where a cell skips reproduction under stressful conditions. So when the density stress is high, the cell will choose not to reproduce and instead just sit there. And so when we image it, it will contribute to the unicell population, but it's not like actively dissociating that generation. We also see, as I mentioned earlier, an approximately 15% multicellularity even after the drop, which might indicate a small niche for colony phototaxis. One of the things that potentially causes multicellularity is the ability for cells to work together to swim. And so even under stressful conditions, density stress conditions, cells might stay in a colony to swim up to the top of the media and get access to light. So it doesn't matter how dense the media is, or the flask is, they will always swim up and maintain around that proportion. The other thing that could be causing this about 15% multicellularity is other kinds of multicell structures to be present at high density, which is important because not all multicellularity that we observe is the same. This figure is from a different experiment. I didn't class the different kinds of multicells in the main one that we did, but this shows that the size of the multicell gets smaller at high population density. Under less dense conditions, we see a lot of 9 to 16 cell colonies, this very classic Gonium plate. But under high density conditions, we see a lot of smaller multicellular structures. They're still classed as multicells, but they do get smaller. And I kind of want to know what's going on with these. It could potentially be just that when the cell divides, it's dividing into smaller colonies. Gonium, as it divides, has a number of divisions that it goes through, which creates a power of 2, so 2, 4, 8, or 16. A 4-cell particle that we see might be a result of a cell just doing 2 divisions and splitting into 4 cells, but that doesn't explain the particles that we see with odd numbers like 3 or 5. So clonal reproduction is not sufficient to explain these small particles. An obvious thing here is that when a colony dissociates, it's not necessarily true that it must dissociate fully into unicells. It could have small groups of cells leaving as a group, or it could leave behind small groups of cells. But another thing I want to introduce potentially is that non-synchronized reproduction can leave behind these small irregular groups as well. And this is actually supported by some microscopy that we did. We do see the classic Gonium reproducing as a group into small groups, as in A and B. The particle that we captured here, C, appears to be a large colony reproducing as a group in a relatively synchronized way. But we do also see particles like D that aren't so synchronized. So some of this is pretty far along in reproducing into daughter colonies. Some of these cells look like they're starting to divide, and some of these cells here look like they're not dividing at all. And this is further supported in 2 pictures taken a couple hours apart of what I believe to be the same colony, where in E and F, E here, we have cells that are dividing and starting to reproduce into daughter colonies, and this cell, which looks like it's just starting to divide with a 1 division across it here. And then a couple hours later, we see that all these cells have started to break away and form daughter colonies, but 2 cells have skipped division this dark phase and will be left behind as a small multicell particle. Realistically, it's probably a mix of multiple things. These small particles.
Just back to that image, please.
Yeah.
Why did you say those 2 cells will be left behind as a small multicell rather than just a single cell? Or I just missed that.
So these 2 cells will continue to stay stuck together. These haven't dissociated, so the mucilage around them hasn't disconnected, and so they'll stay stuck together. When this didn't come through the flow cam because this is on the inverted scope, but if it had come through the flow cam, we would draw a box around it and just look at 2 cells here. And so it would show up as a multicell in our data when we are going to classify it. But it is smaller than the previous colony that it used to be a part of, and certainly smaller than a full-sized colony.
Thank you.
So these small and sometimes irregular multicells that we see could be a mix of clonal reproduction at small division numbers, cells just dissociating in small groups and leaving behind small groups, or non-synchronized reproduction, leaving behind quiescent kind of leftovers. But there's also a potential that there are non-clonal colonies, palmaloids, where cells that aren't necessarily genetically related will stick together through mucilage, or possibly get their flagella entangled. I don't have any pictures of that to show you, but it does happen at very high densities. Another thing that is important in understanding why Gonium is dissociating is to look at the purpose of single cells. Because of the way that the flow cam works, we can't follow up on any of the cells that go through it, but figuring out why Gonium's dissociating is going to be linked to figuring out the fate of unicells after they dissociate. So in a dense culture, the unicells might be dividing unicell to unicell to unicell, which would indicate a reversion of the multicell lifestyle, and that dissociation is a cell-level adaptation. They could just be quiescent, just wait it out until better conditions, and after those better conditions are achieved, they may be dividing into multicellular groups, which would indicate that dissociation is a group-level adaptation geared toward propagule dispersal. There's also the possibility that even in better conditions, they don't do anything, in which case they die, and that's not really adaptive for anything. Or that even in dense conditions, once they dissociate, they reproduce into multicell groups, which would then presumably also dissociate. The level of selection that indicates is not clear, but it does tell us that a single cell is better for reproduction. So they are not going to be able to hit their growth checkpoint and get all the nutrients they need to divide if they were in a group, but they may as a unicell. But again, what level of selection that's acting upon is not clear. Another thing that has come up recently is that media choice might be an issue. Our experiments were conducted in SVM because the Gonium in the culture collection is grown in SVM. But a lot of Gonium literature has Gonium grown in VTAC or SVMA, which is the same as SVM, except that there is acetate acting as a carbon source for the Gonium to grow chemotrophically on. So Gonium is a photosynthesizer, it's an algae, but it also has the ability to use acetate as a carbon source to grow mixotrophically. And this mixotrophic growing on light and acetate might be a standard kind of natural condition, in which case depriving them of that and having them grow only on light may actually be an extra level of stress that we have imposed. This would certainly be interesting, but it would be not as standard conditions, and so another experiment would be important to kind of contextualize that. And we think that acetate would have an effect on the multicellular expression of Gonium, because RLSD expression has been related to acetate, RLSD being a stress response gene that is thought to be related to the regulation of group living. So if this gene that may potentially regulate group living matters, if there's acetate or not, whether or not there's acetate could be affecting how they maintain their groups. So, for the future directions, I want to do an experiment with acetate media to recontextualize our main experiment, and it might end up being that we have given them a stressful condition, but this would still be interesting. We would still find out things from doing this, that in particular, not having acetate is a stress that then would have some kind of effect on group living, which could be interesting. I'd also like to further refine our understanding of dissociation behavior to address the cause of the small cells being maybe fewer divisions, unsynchronized reproduction, or direct dissociation, and importantly address which level of individuality dissociation is adaptive for. And that would be done during, with the cell fate experiment. because this project is an extension of my kind of introductory project, there are a lot of procedural things that could have been done better in hindsight. I'd like to measure turbidity as a measure of cell shading and possibly light stress. Also, measuring chlorophyll would be a better way of analyzing density than just using the particles per mil from the flow cam. And AI automation of particle classification would be very nice to have. That is almost ready, but it is not ready for everything that I've done so far. I also want to get more specific about what stresses are acting at high density. This experiment just shows that high population density has some kind of effect, but if we could figure out what at high population density is important, it could be waste and chemical signals from the media just being too crowded. It could be cell shading and light stress, or it could be that nutrients in the media itself are being used up, and therefore it's too depleted. Doing experiments with depleted media with cells filtered out and diluted media to just reduce the nutrients would help kind of figure out that difference. And that's all really in service of describing what the operative stress is that influenced the cost-benefit change at high population density. The significance of all this is understanding how Gonium handles uncommitted group organization under these stressful conditions. And understanding how Gonium does this could help us better understand the evolution of multicellularity generally. Of course, early group organization being necessary in all multicellular lineages, as simple unicellular organisms evolve into committed, differentiated multicellular organisms. And this will help us better understand the origins and complexity of life in general. Questions?
That was great, Clyde.
Thank you.
Really, really. I just want to say, and all of us feel this in the lab, but since we have a guest here from outside, Clyde, you are a very unusual undergraduate in that, you think creatively, originally, and independently about these issues, and this is something that we value in working with you. And but I just want to say how unusual that is at this stage. So that you are thinking independently. And an example in this talk is this point that you make about the dissociation seems to be leveling off. It doesn't go to zero. So that's something I'd never really appreciated until you said it. Could you had some ideas about why you thought that was the case? Could you just
Yeah.
return to those? I didn't get them all.
Yeah. So some of this is new. I did a kind of side experiment for some other classes looking at phototaxis in Gonium. And in writing up that paper, I kind of stumbled across the idea that the reason we don't see multicellularity go to zero is that groups that are swimming could have the ability to get to the top of the water column in media, even at very, very, very high densities. So if you have a flask that is so thick with algae that light can't penetrate at all, you might still see groups that are able to swim up to the top and achieve enough light to stay in a group, even at very high densities. So that was one thing. And there is some data that I can show about that.
So wait,
as
so
well.
that's your first bullet here, the small niche for colony phototaxis.
Yes.
So the niche, just the ecological niche you're imagining that that is a refuge for multicellularity
Hmm.
is what? It's the just to say it again. Yeah.
So in a really dense flask, there's still going to be an amount of that flask that has direct light access, that being the top and the sides mainly. Uh,
Gotcha.
and so niche is kind of a weird use here because usually we use that to refer to like
Yeah. No,
species,
I
but
like the word. I liked it. Yeah.
because we have such a potential difference in phenotype, and that a multicell colony has kind of different pressures on it if it is multicellular, I think it's kind of appropriate here. So the niche being like the best swimmers are able to access light, even under very, very, very dense conditions. And so
Mhm.
a multicell colony with organized swimming could still achieve that benefit enough to outweigh the costs of highly dense media by swimming really well. But that
And
wouldn't be
can you,
available to the entire population because some of it has to be not at the
right.
top.
So can you think of some experiments that might test that hypothesis?
I did one where I sampled from the center of a flask in dense conditions and from as best I could at the top of the flask in dense conditions. And actually, a further extension of that experiment where in the flow cam um, I let the sample kind of stratify a little bit. I let some of the colonies swim to the top, and then I ran through the very end of the sample and was able to see the multicell proportion in a kind of bubble that had formed above the rest of the sample. So all the good swimmers made it up there and were concentrated at the end of the run. And I did see an increase in multicell proportion at the top of the water column there.
I mean, I was thinking of just the structure of the vessel. I mean, you could do two experiments
Mm,
where you had a tall and very near a tall vessel with just very little surface area of light
that's a
exposure
good
at the
point.
top versus a very flat, or just vary the amount of light, the surface of light exposure somehow
Yeah,
to see
if
what
I had
effect
like a
it
test tube and taped up the sides versus
uh,
like a
But
plate.
like
Yep.
in your big experiment, I mean, don't you have them on a shaker?
Yes.
So I'm just thinking out here, but with now swimming ability, it seems like when we have them on a shaker, the whole idea is to kind of for all of them to now experience now similar environments. So I'm not sure whether swimming really helps when you're kind of on a shaker.
Yeah,
Um,
I think it would certainly reduce the possibility of that. I will note that
advantage.
the
Yeah.
experiment that I did looking at different heights was with a flask that was not on a shaker.
Yeah, no, no, I completely agree that now if they're swimming, they might be able to access the no the. But then, as I was listening to your talk, and I the same as Rick, now I kind of didn't think of know why some of them are still multicell. And again, Rick, you know more about this ecological, but what is that? Can this be explained just as a bad hedging strategy? The idea being that even when you showed how in a colony, there could be a couple of cells that are actually not dividing,
Yeah.
or some you're saying single cells might divide now or not. I was just
mm,
wondering, and I'm just thinking loud, whether this could be a bad hedging strategy. The idea being that if you have in a population both single cells, assuming that that's advantageous now in these dense conditions, and some multi cells, such that if the conditions become worse, no, the single cells are no, that's one no advantage. But if the conditions get better, then you still have some of the multi-cell population ready
Hmm.
to go.
Yeah,
So
I think that's
I'm not
certainly a
sure
possibility.
if that and Rick, would that be an adaptation? So assuming that now somehow this kind of a strategy evolved, that would be an adaptation at the group level, right? So the idea that you would produce two types of propagules, single cell propagules and multi-cell propagules, to be ready for either type of environment.
Yeah, that is interesting. I didn't think of that.
But again, I don't know enough of this. The ecological. What do you think, Rick?
I just kind of missed that, Aurora, about group level and cell level, how it plays into that.
No, I was just asking if this is a bad hedging strategy. That would be a strategy that evolved now at the group level. So basically,
Right.
it makes if the idea is that the presence of the two types of particles, single cell particles, and so you know how Clyde, we talked about the potential. If it's a single cell, maybe it's just a reversal to single cell strategy. So that would be a cell level strategy. Now, if the single cells are actually waiting for better conditions to make groups, that's no group level. If they are dispersal propagules, that's a group level. And I'm thinking maybe there's another possibility in that diagram where the single cells are just part of this bad hedging strategy.
I like that. I'm going to write that down.
And that would be actually, again, another group level adaptation, that now under these conditions, you produce two types of propagules to be ready for, if the environment gets worse, then you still have these single cell propagules as dormant, dormant stages. But if it gets better, you're not starting with, imagine if everybody goes single cells in a dormant stage. When the environment changes, it takes a while to get out of this dormant stage, to divide, to make a multi-cell. But if you already have a small proportion of multi-cells, when the environment changes to get better, then you suddenly can take over you have a head start. What's that expression? So I don't think we thought of this. And again, I don't understand much about, I don't know much about, but to me, it seems like, that would be kind of a cool, especially when you showed us those pictures with some cells, and the idea that maybe the single cells can still make. Yeah. So that one maybe those two cells are not necessarily defective cells, or maybe that's the idea, some cells in the group
That
to
would be
make
interesting.
multi-cells.
Yeah.
Yeah. But I have to say, I did not appreciate, and I want to kind of congratulate you on that. I didn't appreciate, I've always imagined that the single cells end up breaking off the group. But you always made the point, and I didn't necessarily take you seriously initially, you made the point that the single cells might be actually left over. Like, it's not that they're dissociate first, they just leave the group, okay, basically get off the ship, the sinking ship. But actually they are left over.
It could be.
From
so
yeah.
I think
or
that
it could be a combination.
it's
Yeah.
probably a combination of both. There's no way it's only leftovers, because we see too many unicells for that to make any sense. But they certainly might be leftovers.
Interesting.
Question.
Mm-hmm.
Maybe it's sort of irrelevant. Is it known how these buddies sort of perceive and interpret threats?
Like stress?
Yes, stress, any of the stressors, because you talk about, how they respond to, maybe it's not enough light, or, overcrowded conditions. So I'm wondering, and you mentioned at one point a certain gene, so somebody must have been doing research on identifying genes that was like an R, yeah.
Mm-hmm.
And so I'm just wondering if or if anybody knows kind of like what the mechanisms that these organisms use to sense the stressors that you're talking about.
So there definitely are some mechanisms. There's genes that are stress response genes that activate under certain conditions, and also I mentioned that light stress, like cell shading, might be something. They actually have an eye spot that is able to detect light. I'm not sure if that's used in stress response, but it's definitely used in phototaxis. But if somebody else wants to jump in here with a better understanding of some of the molecular side.
And what about oxidative stress, Clyde?
Yeah, so I'm not super big in oxidative stress, but as I understand, metabolism and I think heat stress also causes like stressful conditions can cause buildup of oxygen in reactive oxygen species that are deleterious to the functioning of cells, but then also the presence of reactive oxygen species can then be harnessed as a signal of stress, which I believe is important for some of the genes related to RLSD, going into like RLS1 and then Reg A being. That's not my area. I just.
Well, that's the hypothesis for now, because we see a correlation between ROS production and this gene expression in other model organisms. So in the clammy, that is unicellular, and Volvox, that is differentiated and multicellular. So because we see this correlation, we were kind of proposing, or maybe this is like a universal signal that is important for this gene expression, that indicates stress in all of these different species in the Volvocene algae.
And I'm assuming that I know nothing, so kudos to you for, talking about something. You're talking with like the super experts, and then I know absolutely zero about any of this, and you're having to make it intelligible to both of us. But so I'm assuming that in your experiments, it's like sterile, and there's no other life going on, right?
Ideally.
And I'm
we
Okay.
try
but
very
I'm
hard
curious
to make
about
that the
like
case.
if if these things that they're doing have been adapted to be in competition with other organisms that they must live in the real world around and with all the time.
Well, one thing
How
that
does
we saw is some of the Gonium strains that we had in the culture collection were actually contaminated with something else, and that was causing a reduction in multicellularity, I think. We didn't do a super good experiment about that, but they were having a hard time, and it was because of contamination that was in there. So there are certainly stressors that can be based on other organisms and or competition, not just within its own population, that would cause an effect on multicellularity.
So, for example, Clyde, do you want to talk a little bit about predators and predation, maybe?
Sure. So predation
Character.
actually is an example of a stress that can actually increase multicellularity. The idea here is that a unicell is pretty easy to bite down on if you're someone who feeds on algae. But if there is a colony, especially a large colony, that's going to be a lot harder to get inside your mouth and chew on. And so there are also stressors, predation being the big one, that would cause a selection pressure for increased multicellularity. And I believe there is actually a strain of Chlamydomonas that was experimentally evolved under predation selection to be multicellular in palmoids, which is a kind of aggregation of not necessarily genetically related cells that come together to just stick in a really big clump.
Right. I'm going to have to disappear at 9. So
Okay.
I just want to interject a few other thoughts here. As you look towards the summer, um, so Clyde is going to be working on a research RA this summer,
I need to do the form
and
for that.
just, pardon me?
I need to turn that in. I had to turn it in my proposal.
Yeah. So and that's what I wanted to just focus on. So what I want you to focus on in that proposal is is some of the results um, of your work that you just discussed. So this whole idea of, I mean, the big bullets that I kind of am curious about is, the frequency of multicellularity, that this refuge idea that somehow
Okay.
um, I mean, that's a nice idea. And then there's the idea of, group versus cell level. And it could be related to that question, like Aurora was thinking. Um, and the group versus cell level, that's something that Sori is working on with regard to gene expression
Mm-hmm.
of RLSD. And so there's some synergism there. Um, and then there's this just a general idea of stress that we were just talking about, and you were talking about acetate stress. That well, no, sorry, has hypothesized that the acetate is more stressful to groups, right? I,
Yeah, that's correct because they changed their metabolic strategy of like using the fatty acids that is showing to create like more hydrogen peroxide as a part of its byproduct. And so, yeah, I'm proposing that that may be more stressful.
and so how does that fit into? I mean, Clyde was suggesting that no acetate is stressful, right?
I
So,
I
so
actually disagree. Yeah, I think it might be that the lack of acetate might cause more stress in on net.
Yeah,
well,
I think it's like really difficult to think about like what is stressful because I think there's also this level of is it stressful to be like because there's a difference between the group phenotype being stressful versus a single cell being stressful. Like maybe in SVMA, it is stressful to be under a group phenotype, but then in SVM, it is stressful to be as a single cell. Like I feel like there would be differences again in both of those scenarios, which I'm
I
not sure.
think our issue is now when you say stress, you always know how it's relative to what it's considered to be optimum.
Mm-hmm.
But for us, I
Mhm.
think our confusion right now is that now what's the default? What's the optimum?
Mhm.
Now, if outside in the environment, nature, they have organic now substances, so that would make the SVM with acetate more like the optimum conditions. And in that respect, now Clyde is right that now withdrawing the source of no carbon might make it stressful. But then because now we started from the assumption that the medium without the acetate, the SVM, is the optimum because that's how we grew them, then we kind of anything that was different than the optimum, no, we interpret as potentially being stressful. So I think it's the egg and chicken kind of dilemma. So we're trying to now we need to figure out or no uh, yeah, what is considered optimum? What is the optimum condition for gonium? If we agreed in the environment and the way that initially when people isolated gonium from the nature and they grew them in some media in which they put a little pea and some kind of a, I think, and they are mixotrophic, right? So if they are
Mhm.
mixotrophic, meaning that they can use carbon, that is because in nature they use that. So that's an adaptation now. So the
Mhm.
only way you can explain the mixotrophy, so the ability to use carbon, is that out in nature they use carbon. So then now this implies that the optimum conditions should be with carbon, with acetate. Um, but you might be right in your thinking. Sorry that now acetate is not necessarily stressful, but when you add the density, no,
Mhm.
the increased density and this type of metabolic is the same with our food or something. Now, okay, maybe some food is good now at some level, but the same food, if it's a lot of it, it's going to produce more ROS now.
Mhm.
So it's kind of the levels and the context. So
Yeah, the
as it's probably not
context.
stressful, but now under some contexts can be stressful. So I
Hmm.
think that's what you're trying to say, that now under
Yeah,
the
yeah,
context of now high density, now the acetate may be more stressful.
exactly.
And when you're not using acetate, it's you're vegetarian. I'm just not giving no, maybe in those contexts, no, it's basically more stressful compared to non-vegetarian, but under
Hmm.
crowd conditions, maybe now you don't feel the difference so much. If you're
Hmm.
vegetarian, the increased density doesn't affect you so much. But if you're on acetate, the increased density it makes this metabolism now a little bit more dangerous
Mhm.
if you're in crowded conditions. So I think that's the kind of the point you're trying to make.
Yeah,
Um,
for sure.
which metabolism is becoming more dangerous under crowded conditions.
Mhm.
So I'm going to have to leave. I hope the rest of you keep talking if you're able. And Dee, it's been a pleasure meeting you. I'm sorry we couldn't just chat more. And I know you're working on a PhD thesis, I think, is over in Edinburgh. So I'd like to hear about that. But unfortunately, we don't have time. But Clyde, you did you hit a home run with
Thank you.
this work,
Yes.
Clyde. And we're so happy to have you in the lab and happy that that you're putting your original thinking into this. And so we're looking forward to you figuring this out, right?
Excellent. I am
The
too. I'm
different,
so curious now.
yeah, so the different meanings of stress and how it relates to acetate versus non-acetate and the results that you're seeing here in the group and cell level, and then the why doesn't multicellularity go to zero? I think that's looking
Mhm.
forward to seeing your thoughts on that in your proposal for the summer.
All right, we'll
And
do.
so anyway, it was a really great
Yes.
defense, Clyde, and you deserve your honors
Woo.
for sure.
Yeah.
No question about
Yay.
that. Bye, everybody.
Have a good one.
Bye. Yes, Clyde, I think this presentation was the best you gave. Yeah. And by the way, can we have a copy? I want to
Sure.
think of
Oh
Yes.
some
Yeah.
yeah.
look at the data a bit more. Yeah. But I have to say, I think in this one you were really able to put all the thoughts, together and all the data together in a way that I don't think I no I mean I saw in different presentations, but in yeah, it was a
Mhm.
really nice way to synthesize everything,
That's
both
I'm
from
Yeah.
I'm
experimental
glad
and
that that came through because as I was working on this, kind of just had a lot of ideas and was like, I should throw that in there. And I ended up going, I think maybe even a little more into like the origin of
yeah,
these cells
but
that I'd
no,
never
but that
talked
that
about
was
before, and
great.
it was kind of I
Yeah,
was
no, that
worried that
was
it was going to be a little too new for the thesis part
Mm.
because I would totally
hmm
present this in a lab presentation, but
hmm
my prompt, so to speak, was to make this more general. Um, so I was slightly concerned that that
No, it
would be a
was
little
great
too
because
raw for this, but
it hit on all basically it hit on all aspects. Yeah, so basically it's in
Yeah.
one you managed to address all the points and issues, and yeah, so was and I have to say I'm gonna steal. I mean, I'm gonna make my own, but I'm gonna steal. I love that diagram from the single cell to multi-cell
Oh,
when you
me
made
too.
human from single cell.
I did
So
that. Okay,
I'm not gonna steal it for. I'm gonna spend a lot of time trying to make one myself, so I'm not no.
you
But
could
I'm
use
gonna
this one if
steal
you
that
want.
idea. That was the best
I
diagram
loved it.
for the
I was like,
transition
damn.
to single cell to I've ever seen.
I
Maybe I'll make a flower, but still, that human, yeah, that was the best one.
I did that this morning.
Ah,
I was working on
I
this really
love
late
it.
into last night, and I was like, I need to go to bed. So I just kind of wrote down what I needed to do, and then this morning I was like, I gotta have a figure here. I can't think of anything to add. So I was just like, how
Ah,
could I?
that'll be good, even from no multicellular meetings and stuff. It's such a nice
Yeah. Yeah.
visual. Every time people show no a single cell in a group of cells, transition from multi-cell to no, it's kind of whatever. It's very abstract, right? But yeah, that's what we mean by especially for someone who doesn't necessarily understand biology. What you mean, like it's so visual. Where
Exactly.
many
And you can see it.
yeah, with
I'm
one
I'm
of
glad
many.
that one read. That's good.
Yeah. Okay. So do you have exams?
Not today,
Not
thankfully.
today.
This is my activity for today.
Yeah.
I will in the
Good.
future, but
Yeah. And then the summer it's gonna be just full time.
yep, we'll steam
Perfect.
ahead on that.
Yeah, I'm really curious. Yeah, yeah. Hopefully, So you're gonna redo now. You have to redo it with acetate, right? That's the first
I
thing
would
you want
like
to try.
to, yeah. I would definitely like to. It might end up being kind of scaled back. I'm not sure if, well, depends on if I think the acetate might actually cause a difference between the time points.
Mm-hmm.
And or if we need to double stack this again with the molecular work.
Mm-hmm.
Because sorry, I think you're still doing time points on
Yeah,
LSD,
I
right?
mean, we do have to do a growth curve anyway for SVMA. But for Gonium and Clammy, and I'm already planning to do that. So like it fits in well. But then this time, I really want us to go to stationary phase.
Yeah, I
Mm-hmm.
agree. So
Yeah.
one of the things that I feel like is kind of lacking with this experiment is really showing the tail on this. So
Mm.
we captured the majority of the reduction in multicell proportion,
Hmm.
but I basically said I just claimed that it stays at 15%,
15%.
but that's
Yeah.
not shown
Yeah.
here. I have that from flasks that are like a month and a half old, so that's true. But this
I mean,
experiment
it's
doesn't
true,
show it,
but like, yeah.
and so going all the way to
So, and
stationary would be nice.
yeah, and also collecting the chlorophyll data at the
Hmm.
same time. So I'm just like I'm just running like very high density flasks right now, to get the stationary phase to kind of like know at which multi-cell at which population density we're gonna get stationary phase, at
Hmm.
least with chlorophyll data. So after we get that, we can kind of think about how many days to run into for Gonium.
I so this isn't chlorophyll, but with the particles per mil on the flow cam, I where are my thoughts? Where are my thoughts in my head? I ran the experiment with the heights, right? And that was from the culture collection flask that was like a month and some old.
Yeah.
And I think I was, oh, man, I just want to pull up the run now. But it was under 1 million particles per mil. And so
Okay.
I forget exactly what it was, but like that's around the particles per mil that we're hitting stationary. So if we can work back from however far that is,
Uh-huh.
I need to
Yeah.
go actually get the data to say specifically, but that seemed very stationary,
Uh-huh.
and was also at 15% multicell.
Just in case you use this slide again, you have a typo.
Oh no!
Column is yes, freezed at low concentration.
Oh, I didn't mean to imply that they're religious.
Yeah, exactly, exactly. It's like, oh, that's an interesting strategy. Okay, perfect. So are we having a lab meeting next week?
Yeah,
So
I think that would be great.
Ah,
Yeah.
my
And
lozenge
then
ran out.
yeah, because I gathered the Clammy and Gonium chlorophyll data, at least
mhm,
like the beginning. Like I didn't get to stationary for Gonium yet. Because I'm only at like day 10 today.
mhm, mhm.
But I have like like early. And then Clammy reached stationary so fast.
Fast, yeah,
So
yeah,
that was fine.
yeah.
So yeah, we can go through that data, I guess, next week.
Okay, perfect. Okay, I
All
gotta
right,
go too.
All right.
great. Yes,
Congratulations again. You should
yes,
be really
congrats,
happy,
guys.
very proud, and I'm hoping you're very excited.
I am excited.
And we're really grateful to allow us to experience the same excitement as you.
Yeah.
Or you make us excited, yes, about your project,
Yeah.
which is great. Okay,
All right.
bye
All right,
everybody.
Have a
cool.
good one.
Bye,
Bye.
everyone.
Nice to meet you.
Same here. Bye. Good luck with your PhD.
Thank you.